All right, it is 2 o'clock, so we'll get started. As a reminder, your test will be on Friday. It should be all set up, ready to go. So make sure you read the instructions on the announcements to get the ProctorU browser extension downloaded. To find the test, it will be under course content. You can't see it right now, but you should be able to see it when you log in on Friday. If you can't for some reason, please do email me because that means I did not set it up right. So anything we go over today for lecture material won't be on the test, but I do want to give people some opportunity to ask questions about what might be on the test or anything about the test. So let's do that to begin with. So does anybody have any questions whatsoever about the test? You can ask anything you want. Do you have the opportunity to show your work for math problems? I mean, I think you could show your piece of paper to the screen, but for the math problems, I'm just looking for the answer that we get. Because really nothing's too overly complicated for our calculations that, you know, I'm not going to divvy up the points, like cut it like, oh, you used the right equation, that's +2. It's either you got it or you don't. Because if you use the right equation, you should be good to go. Are all questions multiple choice? The majority of them are short answer. There are a few multiple choice, but no, they are not. So if you've been talking to anybody who took my class over the summer, that was all multiple choice. That was because we didn't have this ProctorU set up. Now we have this ProctorU set up. I can go back to testing like I normally do. So that's usually short answer. Do we have the scheduled test time? Nope. As soon as you click on the link for the test, you'll be walked through some like ProctorU things. And then once you do that, I think it takes like anywhere from 3 to 8 minutes to get like all set up through ProctorU when you click on it. Once you're all through that, time will start. So time is not going while you're trying to like get everything ready for ProctorU. But as soon as you can see the test, time will be ticking down and you have 60 minutes for that. Other than amino acids and the structures, what should we focus on? Anything that we talked about in class, anything that's on the reading guides. I'm the test to see how well you know the information from the first month of classes. Anything that I specifically talked about in depth in class, those were, you should know that. So if we spent like a good amount of time on something in class, I usually ask questions about that. Is there a drawing involved? No, but there are structures involved. So I'm not going to ask you to draw anything. Do you have 50 minutes? You have 60 minutes. Anything but the reading guides. Anything but the reading guides? I don't know what you mean by that. When I went through the test, to make the test, because I already made my rough copy, I said to study anything but the reading guides. When did I say that? Just now? If I said that just now, oh, that must have been, either I said it wrong, which could very well be. Anyways, reading guides are valuable. So the way I made this test is that I went through our lecture slides, I went through the reading guides, and I drew questions from there. I'm not saying that I copied and pasted stuff, but that was my inspiration for making questions. Can you go over a question on test 1 from fall 2019? I can if you tell me what the question is. How many questions are there in the exam? I knew somebody was asked that, and I was just talking to my wife earlier today. We could not figure out for the life of us what advantage that gives a student. Because what if I said like there's 2 questions, but there's like 20 parts? Then that's like 40 questions. What if I said there's 100? Is that going to change how you study? We were trying to wonder that, but there's like, since you asked, there's like 26 questions. Are there questions where you have to list the steps and procedures? You should know how the steps and procedures of our various experiments and tests are run. I'm not saying I'm going to strictly ask you, for this method, tell me how it's run. I might, but there will be questions involving if you know how this process is run, you can get the question right, if that makes sense. Question 8 from test 1, 2019. Let me pull that up. This is my Blackboard song, my Blackboard song, my Blackboard song. This is the song I sing when I go to Blackboard. Okay, test fall 2019, test 1. Question 8. This is a fun question. Let me share it with everybody. There we go. All right, question 8. I have a solution that contains 50% protonated aspartic acid, 50% unprotonated aspartic acid. See picture below. What must be the pH of my solution for this to occur? All right. So let's remember here. If a solution is made out of 50% acid and 50% of its conjugate base, which I'm showing here, right? This is the acid form. This is the base form. 50-50. That sounds like a buffer to me, right? And our buffer equation is pH equals pKa plus the log of base divided by acid. Well, if the concentration of base equals the concentration of acid, that means base divided by acid equals 1. If you take the log of 1, that equals 0. So for this to occur, pH equals pKa. This question was really, what is the pKa of aspartic acid, right? Tricky, tricky. That's the answer. It's the pKa of aspartic acid, the side chain. Anything else? You can go for a couple more minutes. I'm very proud of that question. I thought I was super clever making that. Students in 2019 were not happy. So many people said 7. Can we go over 17 of the same test? Sure. Draw a protein backbone at pH 7. Okay. The protein, the backbone of any amino acid / protein at pH 7 looks like this. NH3 positive, COO minus negative. That's a backbone. That question is getting at, do you know what the term backbone means? Do you know it's supposed to be protonated? Those of you who have looked up amino acids on the internet, you will see that people often draw them as NH2, C alpha, COOH. I, for the life of me, do not understand why people do that. That's wrong. That does not exist. They're charged. They're always charged. How to draw it on the PDF? Well, I'm not going to have you draw anything, right? And in 2019, you would have had a piece of paper in front of you. So what I would do when studying for this is, just draw it on a piece of paper next to you. Just see if you get it right, I guess. Oh, how it was drawn. Oh, how it was drawn. I misread that. I can't read English so well. Let me see what it looks like. Mm-hmm. Oh, yeah, that's because there is no protein backbone on that answer key. I think I remember when I was making this answer key, somebody came into my office and asked me a question. And so I used this as scratch paper. And that's why you see all that KM nonsense. And then I forgot to go back and correct it. So yeah, that makes more sense. For the amino acid, the structures are different in picture to picture in Google. What structure should we follow? You should follow the ones I put up. The only difference on Google are going to be protonated or deprotonated. That's the only difference you will ever see on Google. They could show them in different forms. Like, what I mean by that is like different projections, I should say. But the same atoms are present, the same bonds are bonded the same way. For some reason, Google loves to protonate everything and make everything neutral, even though that doesn't exist that much in nature. But yeah, the ones I gave you should be all protonated and deprotonated. They should be good. Anything else? Anything you desire to know? Anyone? Anyone? Last chance. If we have to list the names of the amino acids, do you prefer it in a certain form or will it specify on the question? Do you mean full name versus 3-letter code versus 1-letter code? If for that, I will tell you on the question what I'm looking for. On future tests, if I ever ask you an amino acid question and I'm looking for a specific amino acid, all 3 names are good. They all mean the same thing to me. If you say C or cysteine or cis, that translates the same to my brain. So, but on this test, if I'm looking, because we are getting tested on the 3-letter code, the 1-letter code, and the full name, I will tell you what I'm looking for when. Anything else? If we have any questions during the exam, what do we do? If we don't understand the question, you can email me. Well, I don't know actually if you can do that, because what will happen is that the program will flag that you opened up something. So I made it like last night at 10 to 11 p.m. For some reason, I decided to make the test then. I'm gonna go over it again after we're done here. I'm gonna go over and take a look at the test. and I'm going to try and proofread it as best as I can. If so, hopefully there won't be any confusions, because I don't try to throw trick questions other than that like one buffer one, because that's a good question. But my goal is not to confuse you. My goal is, as an instructor, I have to certify to the rest of the world that you know what's in this class, right? And how do I certify that? I test you. So it's in my interest to ask the questions straightforward so I can know what you know and what you don't know. So when you get that grade, when I assign you that grade, that is a fair certification. So what I would do is when you're taking ProctorU, there is a help thing for ProctorU that you can ask them help, and they do have my contact information. And if it's something like, I don't understand what this question is saying, I think they will reach out to me and try to give me an answer for that. Keep in mind, on Friday, I do teach between 10 and 11. And if you take it early in the morning or late at night, I'm probably not going to be able to help you. But if you take it during the daytime and you talk to them, they should be able to get to me. Yeah, you can't have other programs open. Yes, that's why I walked back on that email me. But there will be, I believe there's a ProctorU like help me button, which you can contact them and then they can contact me. Pop quiz, what amino acid did I just draw? Who knows their amino acids? Who knows their amino acids? B for Baleen. Since we have this non-cheating thing going on where you're videotaping or getting recorded, this time I have opened up all the questions so you can see them all at once. Normally what I do is usually I would just give you one at a time. Here, we're going to try to replicate a test as much as possible, right? So you're going to have all the questions. One thing I do want to say though, I'm trying to be flexible to people's schedules. So do me a favor, and if you take the test early, don't tell people what's on the test. Because why would you help them out to get a better grade, right? Just keep that information to yourself. I can't enforce that, so I'm asking everybody, please, if you take it early, do not discuss the test until Saturday. When Saturday happens, you can talk about anything you want on the test. But I just ask, please, let's try to keep this as fair. Because like I said, the whole point of the grade is just a certification that says, yes, this person knows what they're talking about, or no, not really. Any last minute questions? I'm willing to go to 2:20 before popping up that PowerPoint. You don't download a PDF. It's not all of it's multiple choice. Some of it will be you type into an answer box, short answer. The majority of the test is short answer. There are multiple choice questions. There's calculations, but the majority of it is you have to answer it like it would be on a test with a pen and paper, but instead, no, you're typing the answer. Pop quiz number 2, what amino acid is that? It won't flag me for looking down at my pen and paper, right? So I'm allowing people to have scratch paper. And I think what you have to do, yes, that's proline. I think what you have to do is make sure your area is clean, and the camera will be able to see that. And then what I'm guessing is that you like show them your paper that is blank. And as long as you're not like moving that paper all around, as long as you keep that paper in front of you and then write on that paper, you should be good. Yeah. Last amino acid. Who's that lovable scamp? Yay, good job, everybody. That is glycine, simplest one. See, biochemistry is not that hard. There's only 20, right? And that's what's going to be the test for that. I'm going to show you a picture and you give me the name. If this was a real class, I would give you the name and ask you to draw it, but you know, we're not doing that. All right. Let's get to our PowerPoint. And if you have any questions about the material from now until Friday, please let me know. Once Friday rolls around, I might not answer questions just because the test is live. So get those questions in on Thursday. Okay. So if you remember Monday, we talked a little bit about purifying proteins. We talked about busting open a cell and all that good stuff. We talked about ELISA. What we're going to talk about today, and we're not going to finish, but just to talk a little bit about, is we're going to talk about how to purify proteins. So those of you who are taking lab right now, some of this we actually already covered in lab, but we're going to go over the whole shebang again. So when you purify proteins, you are purifying them based on their biophysical characteristics. Physical means like solubility, ionic charge, polarity, size. Bio just means they're biomolecules, right? And we have different ways of exploiting these properties. So to exploit a protein solubility, we use the process that's called salting out. I believe you might have done this in molecular cell bio. I'm not sure though. But basically, you add salt until your protein comes out of solution, and then you take that protein and you can redissolve it. Ionic charge, that's when you're using like a column to separate proteins based on their charge. Or you can use what's called electrophoresis, where you take electricity and you separate proteins based on their charge. Polarity, we can use what are called hydrophobic interaction columns. So it's just a column with a bunch of hydrophobic groups in it. And size, size exclusion chromatography, also known as gel filtration. SES page also works. In binding, if it binds to something, we can use something called affinity chromatography or affinity columns. And we're going to talk about all these techniques in depth. To purify a protein, you usually don't use just one step. You use like as many steps as you can. And it's really an art. The first time you ever purify a protein, if you're the first person in the world, you get to figure out all this stuff. You get to spend months of your life just figuring out what is the best way to purify this protein. And the goal is to make your protein as pure as possible. When doing this, you will lose your protein. Whatever you start with, you're going to have less of when you finish. But the goal is to not lose your protein while losing all other proteins. So even though the amount of your protein will go down, the purity of your sample will go up a lot. So for that question about the test, pH is a buffer 12. I will do that super quick. Okay. So I just got a question about doing something for the test, even though it's past 220. The question is, pH of a buffer with pK 12.12 contains 60% base and 40% acid. What's the pH? pH equals pKa plus log base acid. pK is 12.2, log base is 60, acid is 40. 60%, 40%. So you just solve from there. Anything to do with the buffer is the Henderson-Hasselbalch equation. Yeah. So the first thing we're going to look at is salting out. You can also do salting in, but most people do salting out. What salting out is that you add salt, and it's usually ammonia sulfate is the salt of choice. You add your salt. Let me talk about this with the image. So here we have our image, and we have 3 proteins, green, yellow, and red. We want to get green. So what you do is that you add salt until right before green is soluble, or right before green crashes out of solution. And so on the bottom of your test tube, you would have whatever solid there is, it's not green because you're going to add salt, but your target will still be soluble. So again, that's art. You have to do experiments and just see, is my protein soluble in this salt concentration, this salt concentration, this salt concentration? So here we have a question. I'll just answer this, right? I'm using ammonia sulfate to purify a protein Q. Q is soluble in 2 molar ammonia sulfate. How would I use this information to purify our protein? So you add salt until 2 molar. You want to go like right below 2 molar. Because under 2 molar ammonia sulfate, your green protein is still soluble, but the red one wasn't. So the red one crashes out of solution. And what you do is you take the supernatant and you add it to a new test tube. Then you add salt to just over 2 molarity ammonia sulfate. Q is not soluble past 2 molar ammonia sulfate. So what would happen to protein Q is that now it's in an environment where it's not soluble, it would crash out. And so what you do is you separate the supernatant, keep the precipitant, re-add your solution minus the salt, then your protein can redissolve. You have just purified some of your protein. So a lot of times that's the first step to purifying a protein. After you bust open a cell, you use what's called salting out to make your protein fall out of solution, and you capture that and then reprecipitate your protein. Questions about that idea? No, we'll move on. Question 2, or point 2. The next way we can separate proteins is through ion exchange chromatography. So what ion exchange chromatography is that you're using the charge of your protein to separate it from other proteins. And you do this by using a column, and this is column chromatography. So here's our column right here. Inside your column, you have beads. You can have positively charged beads or negatively charged beads. When you have positive beads, when positively charged protein, negative beads, positive beads. When you have negative beads, there we go, you're binding to positive proteins. This is called cation exchange. When you have positive beads, you're binding negatively charged proteins. This is called anion exchange. I'll get to your question in a minute, Annette. Let me just continue with this train of thought. So what you do is you add your sample to the column. Then you add solution to the column. Anything that's highly charged will stick to the column. Anything that's not will come out of the column. To kick the proteins off the column, you use salt, right? So salt are ions, NaCl, and when you add that, they will outcompete your proteins, and your proteins will come off. So here in this image, we have blue and yellow left on the column. Blue will be kicked off by a lower concentration of salt than yellow. So what you do, I know this image is cut off, but this is salt concentration. This is time. You gradually increase the salt concentration over time, and different proteins will elute under different salt conditions. So to better understand how this works, I have 6 proteins here. And I want you to tell me, if we have a cation exchange, what order will these proteins come out of the column? If we have anion exchange, what order will these proteins come out of the column? Well, let me get back to the question. So it says it's soluble at 2 molar. How do you know if it becomes unsoluble if it's more than 2 molar or less than 2 molar? You do experiments where you just see with this protein, when is it soluble? And if it's soluble, right? So you do experiments and you see, oh, at 2 molar, it's still soluble. Let me go to 2.1 molar. Oh, it's not soluble anymore. So that's going to be my target. I can go up to 2 molar and it'll be soluble. When I go over 2 molar, it's less soluble, or it's not soluble anymore. So that's trial and error. Most positives come out first. Yeah, tell me. From the proteins, I give you their isoelectric points, so you can tell me which proteins are much more positive. I give you all the information to give me, I want a rank order for A. Order 1, 2, 3, 4, and 5, 6. Order the way they will come off the column. And elute means like to come off the column. So tell me what order in A will they elute? B, what order will they elute? Hint, A and B should be opposite. So I'll give everybody a moment to do that. You don't know how to start? Yeah, well, PI. What is, how to do this? So PI means when a protein is electrically neutral, right? That's the pH at which something is neutral. Okay, at a pH of 2.9, are there a lot of protons or few protons? Does the solution have a lot of protons or does it have few protons? Let's see how well people remember what pH means. And that says protons, not proteins. And proton is H+, or H3O+, rather. Low pH is more acidic, which means there are more protons. That is what it means. Okay, so at low pHs, we have a lot of protons. So if I'm neutral at 2.9, I become neutral. I have no charge if I'm in a solution that has a ton of protons. What do you think I was? Do you think I was negatively charged? Or do you think I was positively charged? What is going to attract positively charged protons? Is it going to be a negative charge or is it going to be a positive charge? So if I want to bind something that's positive, what should I be? I should be negative. Yep. The lower the pi, the more negative you are. That's what pi kind of is telling you. Pi is telling you how many negative, what's your overall charge, right? What are you made out of? Are you made out of negative groups or are you made out of positive groups? The lower the pi, the more negative groups you're made out of. The higher the pi, the more positive groups you're made out of. That's generally what it means. So with that, that actually gives you the answer now. So I'm gonna clear all this. So cation exchange binds to positive proteins. So they are going to be the last to elute. Negative proteins are going to be the first to elute on cation exchange. Again, the word in front of exchange tells you what you bind to. So I bind to positive proteins really good. So order of elution is most negative to most positive. So that's going to be protein 4, protein 3, protein 1, protein, or sorry, not 3, that's protein 6. Protein 4, protein 6, protein 1, protein 3, protein 5, protein 2. Just most negative to most positive. Anion exchange is opposite. I bind to negative things really well. So the first to elute is the most positive. So that would just be the opposite. 2, 5, 3, 1, 6, 4. Cation exchange, negative beads, I bind to positive proteins, amino acids. Questions about that idea? Oh, that's cool. I didn't know I had stamps. No questions? We will move on to the next topic then. The next is the same idea of ion exchange, but it's called hydrophobic interaction chromatography. Here, our beads are just big old hydrophobic groups. They're either like this hydrooctyl hydrocarbon or like phenol groups. So the same idea, but just using hydrophobic groups now. What order would my proteins elute? And look, I give you 1-letter codes. This is a perfect opportunity to review for the test on Friday. Do you know your 1-letter codes? Do you know what is charged? Do you know what's polar? Do you know what's hydrophobic? You should know if you know what they look like, right? So here's a good review. Basically, tell me what's the most hydrophobic. Tell me the order of elution for beads that are hydrophobic. So who knows their amino acid code? No orders? Got any orders here? What order will we have evolution of these proteins? What's the least hydrophobic and the most hydrophobic? Again, I'll help you out a little bit. These are hydrophobic beads, so I'm going to bind to hydrophobic proteins. So the least hydrophobic will come out first. The most will come out last because the hydrophobic proteins will bind to the column, and they'll be the hardest to kick off. Elution means come off the column. How can we tell which one's more hydrophobic than another? The amino acids. Which amino acids are hydrophobic? Which ones are hydrophilic? How can you determine that? What's their side chain structures? See, there is a point to my madness of having you memorize all 20 amino acids, because then we can just look at a code like this and you can tell me which ones are hydrophobic and which ones are hydrophilic, which ones are charged, which ones are not. I don't have you memorize those just for my own personal enjoyment. What if there are 2 or more hydrophobic amino acids? Great, right? Because this is a protein, they're all connected. So you're just saying what peptide is the most hydrophobic? Which one's the least hydrophobic? Right? It's like, what is more hydrophobic? Something with 4 carbons or something with like 10 carbons? Well, the thing with 10 carbons is more hydrophobic than the thing with 4 carbons. So that's kind of what I'm asking here. Protein 3 is the most hydrophobic. Yeah, protein 3 is the most hydrophobic. So that's going to come out last, right? Just look at the groups. W, tryptophan, hydrophobic. L, leucine, hydrophobic. Phenylalanine, somewhat hydrophobic. Actually, it is completely hydrophobic. Histidine, arginine, charged, not hydrophobic. Tyrosine, relatively hydrophobic. Tryptophan, hydrophobic. Methionine, hydrophobic. GG, side chain is. G itself, kind of hydrophobic. So other than H and R, this just has a ton of hydrophobic groups. Yes. 3, 2, 1. So 3 will come out last. Yeah, which protein has the most hydrophobic amino acids, right? So what will come out first? Well, so we know 3 will come out last. Now we have to decide between 1 and 2. So 2, we have a K, those are charged. Asparagine, polar. Serine, a little bit polar. They have their OH group a little bit. D charged, R charged, S a little bit. C, not really. So polar for sure, polar. Polar. Threonine, a little bit. S a little bit, S a little bit, S a little bit. Compare that with the top. R polar, K polar, A no, G not really, D polar, S a little bit, T a little bit, polar. Whoops, I forgot. I was undo. Polar, or hydrophobic, polar. So W and F are big, huge hydrophobic groups. So 1 is more hydrophobic than 2. So you have alanine, right? In 1, you have W, you have F. Remember, W is tryptophan. That's our double ring. That thing is huge. That's very hydrophobic. F, phenylalanine, it's alanine with a phenol ring, not a benzene ring on it. So again, that's super hydrophobic. No, it is a phenol. That's benzene ring. So 1 is the middle part here. So 2 is the least hydrophobic. 1 and 3. So it goes 2, 1, 3. And basically, yeah, that question is, do you know what amino acids are hydrophobic? And then you're just kind of counting where W's, L's, I, V's, F's, those are super hydrophobic. they are really, really hydrophobic. How does a side chain tell if it's hydrophobic? Well, remember from, I think you covered this in organic chemistry. We definitely covered in general chemistry. Hydrophobic means aliphatic. It means I don't want to interact. Well, water does not want to interact with me. So hydrophobic means a bunch of carbons and hydrogens, not a lot of OHs or charge groups, right? So hydrophobic is aliphatic. I know you've learned that term in organic. I'm pretty sure you use terms like aliphatic. Questions about hydrophobic amino acids, or the hydrophobic column. Again, it's the same idea as ion exchange, except this time we're binding to hydrophobic proteins, hydrophobic amino acids. Do we have to memorize if they're hydrophobic or not? On this test, I don't have any questions to say, is this hydrophobic or not? Moving forward, since I told you to know the side chains, I'm just going to explicitly think you know that. And that, I mean, when we talk about proteins, I'm just going to be talking about the amino acids and expect people to know what the side chains are. So I'm going to be talking about, like, look at this hydrophobic pocket, and you're just going to see like letters, right? So test 1, I don't believe there's a specific question. I had to double check that, but I don't think I have a specific question of, is this amino acid hydrophobic or not? Moving forward, when we talk about proteins, when we talk about enzymes, those terms are going to pop up. So you're going to want to know them. Yeah. The last thing we're probably going to talk about is gel filtration. So the way gel filtration works, so if you gave us an alanine which only has C3 as the side chain, is it hydrophobic? Yes, alanine is hydrophobic because it just has the side chain of CH3. So the way that gel filtration works is that it has beads. And in these beads are little holes, little channels, right? And small proteins can fit in these channels. Big proteins cannot. And the way I like to think about this is like, if there's like I guess let's do a maze, right? In this maze, at the end of the maze, there's cheese, right? And during this maze, there's a bunch of side hallways that lead to nowhere, just waste your time. And there's like a mouse that's tiny and a rat that's large. And I say, let's say that that mouse and rat can understand English. And I say to them, somewhere in this maze, there's cheese. Whoever gets to it first gets to eat the cheese. Well, they don't know where it is. And when they go in the hallway, only the mouse can enter in these side rooms. And it's going to, because the cheese could be in there. So the mouse will just go in and waste its time going into these side rooms. The rat has no choice. It has one path, and it's just going to go to the cheese. That is the basic idea of gel filtration. Small proteins will waste their time going through a bead, going through these extra pathways, while big proteins have to go around them because they have no choice. So in gel filtration, big proteins come out first, small proteins come out last. Just so we know what that means, relatively simple question here. For proteins, gel filtration, what order will they elute? 2, 3, 4, 1. Yep. So biggest will come out first, then 85, 55, and 32. Exactly. Relatively simple concept there. And that ends our lecture for today. That's 2:50. We will cover the rest of this stuff on Monday. Like I said, anything that we cover today won't be on test 1. But are there any other questions people might have about anything at all, now that you've had 30 minutes to think about it? Well, we need a calculator for the test. Let's just say yes. I was going to be a smartass and say, well, you don't have to use a calculator because I don't think the calculations are that tough. But let's just say that you should have one. We have to finish the exam on Friday before 11:59. Yes. It will disappear forever until I make it available to get. It's on an automatic clock. It will become available to you at 12 a.m. Friday morning. It will disappear from Blackboard at 11:59 p.m. I would highly suggest you start it before 11. Highly suggest that, because what could happen is that if you get, like, let's say you start at 11:30 p.m. and for some reason your browser crashes or something happens and you try to get back into the test at 12:05 a.m. on Saturday, you won't be able to see it. So that's a good start. Just asking, I don't have to go having trouble. You don't need any fancy calculator. You just need a scientific calculator, something that can do logs and stuff like that. You should have that for this class anyways. You need a calculator that can do logs, can do exponentials. Do you go over the exam later? I might go over it on Monday. Usually what I will do is I'll probably grade it. That's going to be my fun weekend, grading this. And if there's something like everybody got wrong or a large percentage of you got wrong, I will probably spend some minutes to go over it. Or if you have a specific question about it, then I will go over it. Yeah. But so we'll probably see some of it. Yeah. Any other questions that people are burning to know? I play with my stamps. So if we do good in the study guide, we will be fine. If you can answer everything in the study guide and you understand it, you should do quite well, right? Because the study guide is everything we've covered. And they're long. There's a lot of them. That's why I suggested that the study guides, you should be doing them. Try to knock them out during the week leading up to the exam, because cramming. There's a lot of material we covered. Cramming is not going to be people's best friends. You're not going to retain it for the final exam anyway. So hopefully you've been doing your study guides slow and steady. Anything else? This is looking creepy. Well, if there's nothing else, Billy the Biochem clown wishes you all good luck. If you have questions, again, please feel free to email me until Thursday. On Friday, we will become enemies as you try to battle my test. On Saturday, we'll become friends again, and you can ask me questions. Otherwise, study hard. Good luck, everybody, and see you people on Monday. Take care.